Hello and welcome back to Microbial Concepts. Today we are going to discuss about drug resistant bacteria. Actually, there are a lot of terms related to drug resistance, okay, like MDR, XDR, and PDR. So what are these and what is the difference between these terms? Okay, so that is what we are going to learn today. Okay, so before starting to the topic, don't forget to subscribe to my channel and hit the bell icon so that you will get notified on each new video that I upload. And also you can follow me on Instagram by the same handle, that is Microbial Concepts, right? So let's start. So in one line, if you want to define that what is MDR or XDR or PDR, then here is the answer. So if you get a question for say 1 or 2 marks, you can write this. So first is MDR, that is multi-drug resistance. This means that bacteria or organisms which have acquired resistance to multiple unrelated classes of antibiotics. That means different kinds of antibiotic classes are present and the organism or the microorganism is able to, or the bacteria has gained the resistance to those different antibiotic classes. Okay, that's why it is termed as multi-drug resistance. Okay, these classes are not related to each other. Then next is XDR, which means extensive drug resistance or extensively drug resistant bacteria. Extensively drug-resistant bacteria or XDR bacteria are the type of multi-drug resistant organisms that are resistant to almost all or all approved antimicrobial agents. Okay, means they are one step ahead of multi-drug resistance. They are resistant to almost all antimicrobial agents. Okay, so this is the difference here. Then third is PDR, that is pan-drug resistance. So it means that the organism or the bacteria is pan drug resistance. It means that it is non-susceptible of bacteria. Sorry, PDR is the non-susceptibility of bacteria to all the antimicrobial agents in all antimicrobial categories. Okay, so none of the drug is able to kill or is able to act on that particular bacteria. Okay, so this is the difference. Now PDR is one step ahead of XDR. Okay, so this is the relation, or this is the difference between these drug resistance. So there are actually many mechanisms by which bacteria acquires or gains the antibiotic resistance. Okay, there are efflux pumps or immunity or bypasses like tetracycline, trimethoprim, sulfonamides, etc., etc. Then antibiotics, they target cell wall, right? So there are some enzymes which are produced by bacteria like beta-lactamases, which act on these antibiotics and they break down that particular antibiotic. They inactivate that antibiotic so that the antibiotic will not work on the target. Okay, then the targets are modified. Okay, the bacteria itself, it modifies the targets like one like rifampicin or penicillins or vancomycins they act on. Okay, and inactivating the enzymes is the another mechanism. Okay, so examples are beta lactamases which inactivate the beta lactams. Okay, so that is the example. So here are these two simplified diagrams or pictures that you can refer and you can help, or you can use these to memorize what are the mechanisms. Okay, what are the examples, right? So in the first one you can see that there is efflux given, immunity and bypass, then target modification, inactivating enzymes are present or they are produced. In second, you will also find the same, but here the picture is a bit illustrated. You will see where the flux pumps are present. Okay, then the modified targets like the cell wall proteins, they are modified. Okay, then alteration of the drug target, so there is a target which is modified, right? So this is a very simple and helpful diagram of pictures that you can use to memorize things. Okay, then yeah, now beta lactamases. Okay, it is actually a vast topic. I will upload another video separately for types of beta lactamases. Okay, so for now just remember that these are enzymes which are produced by bacteria that provide multi-drug resistance to beta-lactam antibiotics such as penicillin, cephalosporins, cefamycins, and carbapenem. So they actually act on the penicillin, that is beta-lactam ring, okay, lactam ring, and which is shown in green color here. And the enzyme, it breaks the bond, okay, and thus it will convert, or it will alter the antibiotic structure. Okay, so now you will get, after penicillinase, that is beta lactamase, it acts on the uh, penicillin, you will get inactive penicillin. Okay, so now this inactive penicillin is not able to work properly, it is not able to target the cell wall. Okay, so that is why these enzymes are highly produced by many bacteria, and they have now started gaining resistance to these antibiotic categories. Okay, so beta lactamases provides antibiotic resistance by breaking the antibiotic structure that we had seen. The lactam ring is broken down. So these antibiotics all have a common element in the molecular structure, a 4-atom ring, which is known as beta lactam. Okay, so the organism which is able to produce beta lactamases is like uh, what we can say, a plus point that bacteria has, because now the bacteria is able to gain resistance to all the beta lactam antibiotics. Okay, so he is having that extra power. Okay, so these are some mechanisms that you should know if you are studying multi-drug resistance. So now we will see a bit detail about these topics, okay, or these terms. So first is MDR. In short, it is written as MDR, multi-drug resistance or multi-drug resistant bacteria. So we had seen that they have acquired resistance to multiple unrelated classes of antibiotics. MDR organism of greatest concern, they include methicillin resistant Staphylococcus aureus, MRSA, then vancomycin resistance Enterococcus, VRE, drug resistant Streptococcus pneumoniae, that is DRSP, and MDR gram negative bacilli. Okay, like Pseudomonas, Acinetobacter, then Klebsiella, Enterobacter, and other species. Antibiotic resistance in gram negative bacteria is widely studied. Okay, and the infections with resistant gram-negative bacteria are a significant problem, causing high mortality and morbidity in the critically ill patients. Okay, even in hospitals, you can easily get a sample of a antibiotic resistant gram-negative bacteria. Okay, because there are high cases found of gram-negative bacilli or gram-negative bacteria having antibiotic resistance. And if you want to study that, just contact just have permissions from your college or institute, and you can contact the nearby hospital. You can get that uh, culture, and you can study that. Okay, or you can um, design, or you can have a aim of working with drug resistance. Just you need to follow all the precautionary measures. Okay, preventive measures. You should have all the facilities to work with these antibiotic resistance, because they are highly infectious, and as they are drug resistant, you are having high chances of getting infected as well, because you're working with that, you're going to work with that. Okay, so if you are handling such kind of resistant cultures, then please be cautious and use all the preventive measures. You should have a protocol. Your college or your institute should have all those facilities which will support you for the study, for the research you want to do on drug resistant bacteria. Okay, but you need to follow all the guidelines. Okay, have permissions from your institute, college, or that particular hospital, okay, who is giving you the culture for studying purpose, right? Yeah, so as we had seen, the infections are high, the mortality morbidity rate is also high in ill patients, right? So the commonest mechanism of resistance includes uh, production of beta lactamases, which hydrolyzes the beta-lactam antibiotics and produces and production of carbapenimes. Okay, this is also enzyme from beta-lactamase category, okay, that we will see later in another video. So increasing resistance has been noted to fluoroquinolones and aminoglycosides as well. Okay, not just beta-lactams antibiotics are or the bacteria are getting resistance to beta-lactam antibiotics. There are other antibiotics such as fluoroquinolones and aminoglycosides. Okay, these are the other categories to which resistance is also increasing. In view of the increasing resistance to conventional antibiotics, uh, tigecycline and colistin was used to treat or was used as a treatment strategies, but now resistance has developed to these as well. Okay, so what is done if to first line antibiotics the resistance is gained by bacteria, you use second line, but if there is resistance for both, then you go for the other options. So before or in some cases recent years, tegacycline and colistin were used in combination to treat these antibiotic resistance, but now bacteria has gained resistance to these as well. Okay, so how? So there is another term, that is ESBL, that is extended spectrum beta lactamases. They are produced by Enterobacteriaceae and they can be treated with uh, pepracycline, uh, tazobactam or carbapenem like meropenem. Okay, so lot of mechanisms bacteria are using, and now we need to develop, we need to uh, search for a good antibiotic or another options by which we can target these drug resistant bacteria. Okay, so lot of a time you will find that a combination of drug is used. Okay, the synergistic effect is good in some of the combinations of antibiotics and they work very well in case of these resistant bacteria. Okay, so that is also a point that we will study in beta lactamases. Okay, another video which I am talking about. The next is carbapenems producing Enterobacteriaceae is also an increasing concern. For management of these bacteria, colistin and tigecycline seems to be the only option. Okay, C, colistin and the tigecycline, it seems to be the only option. However, tigecycline cannot be used for bacteremia or urosepsis, as tigecycline is a bacteriostatic drug. Okay, so you cannot just use tigecycline as it is a bacteriostatic drug. So while treating such kind of antimicrobial resistant uh, infections, you need to study, you need to know how or by what way the antibiotic it works. Okay, it is a bacteriostatic or not, and the single antibiotic will work, or you need a combination to have a good effect, or you have to have a good treatment. Okay, so a multi-invention strategy is essential to control the spread of antimicrobial resistance. Okay, so if you work in a hospital, or if you know someone in hospital, just talk to them about how these antibiotic resistant bacteria are tested. Okay, how the testing is done, how the diagnosis is done, that yes, the infection is of streptococcus, then is it drug resistant or is it susceptible to the first line second line drugs as well, how it is tested. Just talk to someone that is working in hospitals in diagnosis or testing center. Okay, they will give you a very good idea. Then next is extensively drug resistant bacteria, that is XDR. Okay, uh, many times it is also written as extensive drug resistance. Okay, so extensively drug resistant bacteria are the type of multi-drug resistant organisms that are resistant to almost all or all approved antimicrobial agents. Okay, so they are one step ahead of multi-drug resistance. This term was actually coined in response to Mycobacterium tuberculosis, okay, which was resistant to the first line agents, that is isoniazid and rifampicin. fluoroquinolone and one or more second line parenteral drugs like amikacin, kanamycin, and capreomycin. Okay, this was later expanded to include other bacteria that were resistant to most standard antimicrobial treatments. Okay, so the first it was observed in case of Mycobacterium tuberculosis, but later this term is been getting used for other bacteria as well. The difference is mainly interpreted as to what extent the bacteria are drug resistant, with multi-drug resistant bacteria being unsusceptible to one or more antimicrobial agents and pan-drug resistant bacteria being unsusceptible to all microbial agents. Okay, so just comparing with these other terms, it is interpreted that whether the culture is multi-drug resistant or extensively drug resistant or pan-drug resistant. Okay, so if XDR bacteria are present, the patients are treated and the patients are treated insufficiently, then treatment can cause increasing levels of resistance to develop. Okay, because now the bacteria is one step ahead as compared to MDR, so he is having some powers or some good points in hand to get tolerated to the drug or at least to get resistant, okay, to develop resistance if the treatment is insufficient. Okay, so this was about XDR. So some combination of drugs have been discovered that can be used to treat XDR. Okay, see, and again the point says combinations of drugs. Okay, so one of these is neuroleptic thioridazine. Okay, so this drug in combination with antibiotic agents has shown success in curing XDR tuberculosis when administered by an experienced physician. Okay, so these points are referred from a research article. Okay, so a lot of research is going on and it is observed that in combination with antibiotic agents, this drug that is neuroleptic thioridazine, it works best. Okay, so thioridazine works by disrupting calcium inside the cells. Okay, so this is the mechanism simply. So completely drug resistance TB case was actually first reported in 2007 in Italy. Okay, completely drug resistance. So WHO standards now says that at least 4 drugs are required to treat TB in order to avoid further resistance. Okay, so if we do not work now, if we do not focus on these drug resistance now, then it will be a very big problem in future. Okay, so at least start treating with 4 drugs if the patient is having a drug resistant tuberculosis, mycobacterium tuberculosis. Okay, then next is pan drug resistant bacteria, that is PDR. So PDR is the non susceptibility of bacteria to all microbial agents in all antimicrobial categories. Okay, so PDR gram-negative bacteria, namely Acinetobacter baumannii, then Pseudomonas aeruginosa, and Klebsiella pneumoniae. They are widely spread in clinic due to their strong viability, colonization occurrence rate, and antibiotic resistance. Okay, so as I told you, just if you are uh, having contacts in hospitals, then you can get an idea. If you talk to the person or a doctor, the person can give you a good idea about how these diagnoses are done, what is the rate of having drug resistance cases in the hospital, okay, how they are treated, what are the protocols they follow while handling these drug resistant bacteria, okay, or cultures. So this is a very good topic to even have a thesis on. You can work, you can aim for a particular topic from these drug resistance, you can study for that. Okay, just follow protocols, have permissions from all the authorities. Okay, so because PDR bacteria easily adopt resistant mechanisms, the controversy surrounds antibiotic options with regard to the effectiveness and susceptibility of the resistant strain in clinical practice. Okay, so as PDR, they are highly adaptive, or they can easily adopt the resistant mechanisms. So the options for treating these cultures are very less, as you don't know whether the combination will effectively work and will it show susceptibility or not. Okay, so thereby it is difficult to define a standardized treatment regime against PDR bacterial infections in patients. Okay, so this was about pan drug resistant bacteria. Next is about types of beta-lactamases. So I will just give you an overview on functional classification for beta-lactamase enzymes. So there are actually four groups. First is cephalosporinase, second is penicillinase and cephalosporinase, and third is metalloenzymes, and last is just penicillinase. Okay, so we will study these in upcoming video. Okay, and another point is they are not inhibited by clavulanic acid. Okay, so this is also used in combinations for treating these beta lactamase enzyme producing bacteria. Okay, but these enzymes, they are not inhibited by clavulanic acid, so you need to have another strategy for treating these. Okay, so everything we will study in another video. So we are done with the drug resistance. Here we had studied three different terms. Okay, and I had told you about the difference and how much uh, infectious each one are. Okay, so thank you for watching. Do like, share and subscribe to my channel, and don't forget to follow me on Instagram, okay, by the same handle, that is Microbial Concept. Okay, till then, bye bye and keep learning.